Hantaviruses carry a staggering human mortality rate, killing up to 40% of those infected. Yet they are entirely non-cytopathic. They infiltrate, multiply, and exit, leaving the host cell entirely intact. Taxonomically, hantaviruses belong to the order Bunyavirales, a massive group of negative-sense RNA viruses. Within that order, the Hantaviridae family represents a strict evolutionary anomaly. While nearly all other bunyaviruses rely on arthropod vectors like ticks or mosquitoes for transmission, hantaviruses are strictly zoonotic, maintained persistently within populations of small mammals, primarily rodents. Human spillover requires no bite. Transmission occurs through the inhalation of aerosolized viral particles shed in rodent urine, feces, and saliva. To understand how an aerosolized virus kills a human without directly destroying a single cell, we have to look at its highly specialized genetic architecture. The hantavirus virion is a pleomorphic, lipid-enveloped sphere. Inside, its genome is modular, divided into 3 distinct segments of single-stranded negative-sense RNA, small, medium, and large. The small, or S segment, encodes the nucleocapsid, or N protein. This structural protein coats and packages the viral RNA to form ribonucleoproteins, protecting the genetic payload. The medium, or M segment, codes for a glycoprotein precursor. Host cellular proteases cleave this precursor into 2 mature surface glycoproteins, GN and GC, which form the lattice of spikes on the viral envelope. Finally, the large, or L segment, codes for the RNA-dependent RNA polymerase, the critical enzyme the virus requires to replicate its genome. This tri-segmented toolkit is engineered for one specific purpose: to breach human microvascular endothelial cells and seamlessly co-opt their machinery. The breach begins with a highly specific attachment. The viral G and GC surface spikes bind exclusively to beta-3 integrin receptors located on human microvascular endothelial cells. Once anchored, the host cell actively brings the virus inside by folding its own membrane inward, a process known as clathrin-mediated endocytosis. This diagram outlines the entirely cytoplasmic life cycle of the virus. Because it carries its own polymerase, hantavirus never needs to enter the host cell's nucleus to replicate. Inside the endocytic vesicle, the environment gradually becomes more acidic. This low pH triggers a conformational change in the viral glycoproteins, forcing a membrane fusion that releases the viral ribonucleocapsids directly into the cytoplasm. To begin transcription, the viral polymerase executes a process called cap snatching. It cleaves the 5' caps from the host's own messenger RNAs and uses them to prime its own viral transcripts. This effectively camouflages the viral RNA from early detection. The newly synthesized glycoproteins are processed in the rough endoplasmic reticulum and the Golgi apparatus. Nascent virions assemble and bud into the Golgi cisternae before being shuttled to the cell membrane and released via exocytosis. At this stage, the virus has infiltrated the cell, multiplied, and exited, all without causing cellular lysis or triggering immediate innate alarms. Which raises a mechanical question. Why doesn't the host cell's internal detection system instantly recognize this massive RNA replication event and trigger an antiviral state? The answer lies in a primary virulence determinant found exclusively in pathogenic strains of hantavirus, the cytoplasmic tail of the GN protein, known as GNT. In a normal, healthy innate immune response, the detection of foreign viral RNA activates an enzyme complex called tank-binding kinase 1, or TBK1. This complex phosphorylates the transcription factor IRF3, which then travels to the nucleus to induce the production of type 1 interferons, the critical early warning signals. Pathogenic hantaviruses block this pathway. The viral GN-T protein physically binds to the TBK1 complex, wedging itself into the machinery to create a mechanical blockade. This binding entirely prevents the phosphorylation and nuclear translocation of IRF3. The signal dies at the TBK1 complex, and type 1 interferon reduction is silenced completely. By blinding the host's early warning system, the virus buys itself a silent window to replicate to massive titers. In doing so, it sets a trap for a severely delayed and ultimately catastrophic immune overreaction. Eventually, the sheer viral load overwhelms this stealth mechanism. When the host detects the infection, it triggers an overwhelmingly disproportionate innate and adaptive immune response. The infected endothelial cells begin broadcasting extreme distress signals. They dramatically upregulate surface adhesion molecules like ICAM1 and secrete potent chemokines like interleukin-8 into the bloodstream. Neutrophils are the first to arrive. Drawn by the chemokines, they undergo netosis, programmed cell death, and release pentraxin-3, drastically escalating the local inflammation. A secondary wave quickly follows. Activated CD8+ T cells and macrophages arrive at the site, flooding the local microenvironment with extreme concentrations of tumor necrosis factor alpha and vascular endothelial growth factor A, or VEGF. This flood of VEGF forces a specific molecular failure. It causes the host cells to downregulate VEG-cadherin, a crucial adhesion protein that normally binds the adherence junctions together. At the tissue level, the tight junctions between the endothelial cells physically separate, destroying the integrity of the vascular barrier. Hantavirus does not kill the host directly. It weaponizes the host's own hyperactivated immune system to dismantle vascular barriers from the outside in, resulting in lethal plasma leakage into surrounding tissues. The specific organ system that fails due to this vascular leakage depends entirely on the geographic lineage of the infecting viral strain. New World hantaviruses include strains like Sin-Nombre and Andes virus, which are endemic to the Americas. These New World strains exhibit strict tropism for the pulmonary endothelium, causing a clinical condition known as hantavirus pulmonary syndrome, or HPS. In HPS, the massive vascular leakage causes rapid, non-cardiogenic pulmonary edema. Plasma floods the alveoli, leading to severe hypoxia and systemic shock. Old World hantaviruses include strains like Hantan, Sol, and Pumala viruses, found primarily in Europe and Asia. These strains target the renal endothelium, causing hemorrhagic fever with renal syndrome, or HFRS. In HFRS, the vascular leakage manifests as acute kidney injury, intense internal bleeding, and profound fluid imbalance throughout the body. Whether the patient suffocates in their own leaked plasma or suffers total renal collapse, the underlying mechanistic cause is identical: pathogen-directed immune sabotage. Hantavirus is a masterclass in molecular subversion. By combining a targeted integrin entry with precise interferon suppression, it turns the human body's most powerful defense mechanisms into the exact